In Standard 3, we're not only talking specifically about platelets, we're talking about a process called hemostasis, which is a fancy name for clotting blood to stop bleeding. Your standard reads, I can define hemostasis and explain the mechanisms that help achieve it, and this information is covered in section 14.4 of your textbook. Again, we talk about how blood is a transporting vital substance. However, this cellular component, platelets, is not going to transport anything. It is going to make sure that you do not lose this substance. Because blood is flowing and it's all over your body, any sort of trauma or damage can cause internal or external bleeding. That is a very quick way to have a low blood oxygen count and then to die. So platelets' job is to stop bleeding if there's damage to a blood vessel. Your heading today, BLOOD.3, hemostasis. When we talk about platelets, we're talking about thrombocytes. So we had erythrocytes, which were red blood cells, leukocytes, which were white blood cells, and now thrombocytes are platelets. Platelets are not tiny cells. They're cell fragments, so they're pieces. And they're capable of sticking together to stop bleeding in a damaged blood vessel. That process, the stoppage of bleeding, is hemostasis. There are 3 steps to hemostasis, and we'll go into them in detail. A vascular spasm, a platelet plug, and then blood coagulation. So first, when there's a break in the blood vessel wall, so we have a tear right here, the first thing that your body is going to do is attempt to slow the bleeding. And it does that using the tiny smooth muscles that are lining your blood vessels. So we sense the break, and those muscles will contract, causing the blood vessel to narrow just a little bit. Kind of like when you put a bend in a garden hose. If you can apply pressure, then you can slow the amount of liquid that is exiting through that break. That's what happens first, vascular spasm. That's an immediate response that takes place inside your body, and then we're going to follow it up with the thrombocytes. So when that blood vessel breaks, there's collagen that is lining it along with that smooth muscle. When platelets see collagen, they stick to it. Now that's important because if platelets stuck to each other just in your blood, you would get blood clots and have stoppage of bleeding all the time. So there has to be a trigger. The trigger is the presence of collagen. Once platelets see that and they start sticking to it, then they change their chemical composition a little bit and become sticky to other platelets. And so then we start forming this plug. The plug is a temporary patch. It's going to stop the loss of blood flow, but by no means is that a fixed problem because now we have a totally stopped blood vessel. We just have a temporary plug for now. In step 3, our blood will coagulate. So this is when we're going to fix the problem. This is triggered by blood cells touching outside tissues. The cells of blood can tell when they're inside a blood vessel and when they're outside of a blood vessel. So once they're outside, they'll start to change. Damaged tissues are going to release a hormone called thromboplastin. And that goes through a very complex chemical cascade. There's almost 20 different factors that have a step here. But ultimately what's happening is that there's a precursor molecule in the blood vessel called fibrinogen. When the damaged tissues release thromboplastin, fibrinogen starts to elongate and turn into these long fibrin threads, which are capable of sewing up the damaged vessel wall. They're going to form a new lattice and a new shape to cover up that damaged break. This will form a blood clot, and as a little bit of this cascade and this lattice goes up, it triggers more and more and more lattice to build up. So this is one of the few cases of positive feedback that we'll see in anatomy and physiology.